Thank you. Hi, good morning, everyone. My name is Josué, and I'm going to present you the work that I developed during 2 years at the National Institute of Genomic Medicine in the supercomputing department. This is entitled Integration of a Pan-American Whole Genome Dataset from Publicly Available Data. First, I want to mention that this work and the results that I will show you are part of a publication that were recently published in the journal Diversity and deals with the review of 56 articles where the Native American population was studied by genomic technologies such as sequencing and microarrays. And well, this work allowed us to locate publicly available data. Now, I will now tell you about the presence of the Native American population in the continent of the Americas. Our review, we found in our review that there are a total of 27 countries on the continent that are home to Native American groups, and there are an estimated number of 37 million Native American individuals in these countries. These numbers indicate that there is a large Native American presence on the continent. If we look at the map on the left side, we see that we see the countries with more than 1 million inhabitants, among which Mexico stands out in the first place, next to Peru, USA, Bolivia, and etc. Now, in addition, our exhaustive review work allowed us to locate countries in which there is still not enough genomic representations of many native groups, as shown in the figure on the right side, among which Brazil stands in the first instance out in the first place with more than 200 unrepresented groups. However, countries with few represented native groups also stand out, such as Dominica, Guatemala, or Costa Rica, where there are few individuals that represent an opportunity to be studied by genomic technologies and expand the genomic knowledge of the continent. The next thing that I want to tell you about it's our methodology. In the first place, we used search technologies mediated by artificial intelligence. Specifically, we used the Research Rabbit tool that allowed us to locate all the papers in which native groups were studied with sequencing or microarrays technologies. Then we did a review of these articles. We located the groups, the native groups studied, and the links to public, and we downloaded these public genomes and other data to integrate all this information into a dataset that is publicly available to anyone in a GitHub repository. And finally, we have initiated the download of publicly available genomes of Native American groups for future bioinformatic analysis. Now, this review work, as I already mentioned, allowed us to locate, integrate, and start downloading publicly available genomes to explore the genomics of native groups in the continent. However, the amount of public native genomes is scarce. There are only 93 individuals with genomes stored in public repositories. And this represents only 11 native groups in the entire continent. And the map on the left side shows the countries to which these groups belong, including Mexico with 42 sequenced individuals, Bolivia with 25, Brazil with 20, Peru with 3, Colombia with 2, and Argentina with only 1 individual sequenced. And in the figure on the right side, we represent the native groups with their individuals with sequenced genomes. And I want to emphasize that the native group with more public genomes is the Aymara, belonging to Bolivia. The last one is Chane from Argentina, and the Maya and Pima represent 35 individuals with available sequenced genomes that belong to Mexico. Well, one result that we would like to present to all of you is the types and size of the genomes of some native individuals. So today we have downloaded 65 genomes of native individuals, and of these, 45 are of gram type that represent 1,480 gigabytes, almost 1.5 terabytes, and 20 genomes that are in FASTQ format with a size of 154 gigabytes. That is more less than 200 gigabytes. And well, this difference in file size is given by the content of the files. We need to remember that the genomes in gram files contain the reads of a FASTQ plus the information of where each FASTQ read maps to a reference genome, and the FASTQ file only contains, well, the reads output from a sequencer. Now, why do we need more genomes? Specifically, why do we need more Native American genomes? Well, in short, because the NGS technologies allow us to know the whole genomic context of an individual, and this is the starting point for other research. We can explore genetic variation between individual members of a single species, or we can explore genetic differences between the cells of a single individual, or all this data provides a windows into diverse processes in cell. Okay. Uh, well, finally, I want to mention that this genomic research in Native American populations needs to benefit and have a lot of respect for the individuals. And this is why bioethics should have great participation because, well, the genomics project in Native American population should benefit and respect their participants, and well, clear protocols for sharing genomic data, well, should be established to promote genomic research in these populations. Well, that's all from my part. I thank from this time to my researcher friends who were part of this work and who contributed in a great way. And well, finally, I also thank to all of you, the audience and organizers, for allowing me to show you this work.
So thank you, Josué. Is there any question? Yeah, bueno, I have one. Which was the criteria you decide to they are natives?
Okay, the criteria we defined was in the articles that we collected. We checked that in the article mentioned says that the individuals studied were Native Americans, and the technology used in the articles were using genomic technologies such as whole genome sequencing and whole exome sequencing, sorry, and also microarrays.
Okay, thank you.
Okay, let's thank Josué.
Thank you.
I hope all the people who's, or do you have a question? No? Okay. Yeah, there's one more question. We have time.
Right.
Hi, Jose. I would just like to know what are your future plans with all this data that you have collected now? What's the research you're ongoing with this?
Sure. Well, in my laboratory, we are doing currently some bioinformatic analysis using these genomes to explore like a microRNA variation, and also we are doing selection signals in these native genomes. So we are we are hoping to publish this research in the future.